Hello, everyone, and welcome to this week's edition of Extracellular Vesicle Club. This is a weekly event of the International Society for Extracellular Vesicles. My name is Bonita Powell, and I'm here with Olesia Gololobova. We're both members of Kenneth Witwer's laboratory, who is the president-elect of ICEV. And today we will be hosting and moderating today's session on MRI-trackable EV as SARS-CoV-2 mimetics. And this work was recently published in Nano ACS. So today's presentation will be by Andrea Galisova from Amnon Bar-Shir's lab at the Weizmann Institute for Science in Israel. So if you have any questions, please feel free to leave them in the chat. And as usual, we will get to all questions at the end of the presentation and have a stimulating discussion. Thank you.
Okay, Andrea, I think you can start. Thank you all for coming. For me, it's exciting because the host is Johns Hopkins. I was there for 5 years. It's always good to see some names on the screen. And my lab is doing molecular and cellular imaging. And when Andrea joined us maybe 4 years ago, we had this dream, and the corona changed a bit our dreams. And Andrea will show you what did we do with it. Thank you for coming, Andrea. Please.
Yes, sure. Thank you very much for the introduction, Amnon. So I will try to show you how we try to follow our dreams. So first of all, I would like to thank for this opportunity to share our work here in this amazing EV club. And so I will talk today about genetically engineered MRI-trackable extracellular vesicles, which we used as SARS-CoV-2 mimetics. As it was already mentioned, like this work was already published recently in ACS Nano. So if you are interested in some more details, like you are very welcome to take a look. So I will start with a little introduction about targeted delivery system, because the medical research community already developed many interesting tools for study and treatment of several diseases. But in order to maximize their effect and minimize their side effects, it's very important to have them specific. And in order to do so, for specific delivery of this system, they can be used for example, for delivery of therapeutics with minimal side effects, or for precise activation of biological cascades in cells. Or it's very advantageous if they can be also monitored and imaged either by some real-time imaging or by some non-invasive methods. And we can say, like taking it together, that a good targeted delivery system should fulfill 3 main criteria. As it's reliable targeting, so it has to be specific for some specific tissue or a target. Then versatility, what means that we can use one approach in order to target several targets, using without any extensive redesigning. And also visualization is very advantageous. And as we all know, like we are using these targeted systems, which are extracellular vesicles, and they are natural vesicles which are released by all cells. And we know that they serve for communication between cells, and also they are able to deliver cargos through the surface peptides which they are present on the surface of these vesicles. What is important to us is that we can modify this ability, and we can modify extracellular vesicles or the parental cells by a gene of interest. And then we can, after genetic modification of the cells or the extracellular vesicles, we can produce vesicles which contain specific peptides of the interest. And moreover, we can also load these vesicles with a cargo of interest, for example, by an imaging probe or by therapeutic agents. And these loaded EVs then can be delivered to a tissue of interest by specific binding to receptors on the cell surface. And this starts a cascade of processes afterwards, or also by releasing of these cargoes inside of the cells. To have a good EV targeted system, as I mentioned, it has to fulfill a few criteria as a targeting, very specific targeting. Then it should be versatile, and it should be able to visualize by imaging method, and EVs have all this potential. And if you are looking at this sketch, you can already see some similarities of the images which we were looking at in the last 2 years and what is a big similarity between EVs and viruses. So the viruses also bind to the targeted cells or the recipient cell by peptide-dependent binding or protein-dependent binding. And in the case of coronavirus, SARS-CoV-2 virus, it delivers a viral RNA specifically to the ACE2 expressing cells. And this is the initial step of infection. So therefore, we wanted to contribute to this and to study viruses. And we asked our question ourselves, whether we can use EVs to study viruses, maybe because they share the similarities, or maybe by using EVs, whether we can understand the properties of viruses and their interactions better. So we took a closer look at and compared coronaviruses and extracellular vesicles. And you can see that they share many similarities, as is a similar size, around 100 nanometer. They both have a phospholipid bilayer. They bind to the recipient cells by the peptide-dependent process. And what is the difference between these two systems is that the coronaviruses contain viral RNA, which is infectious, in contrast to EVs, which are non-infectious. On the top of these properties, EVs also have another advantageous properties, as they are genetically modifiable, they are trackable by imaging, and we can load them with the cargo of choice, for example, by imaging probes or therapeutics. So our hypothesis was that we can mimic viruses using non-infectious EVs, and then we can study them by imaging. And how we tried to reach this goal was that we engineered EVs, which had a part of the spike protein of SARS-CoV-2 virus on the surface, only this receptor binding domain, which is smaller. Therefore, these engineered EVs bind to the ACE2 receptors, similarly as the real virus. They share the similar size, so the biodistribution in the body is similar. And moreover, what is important, they don't contain any infectious material, and therefore they are safe to work with. Moreover, we can also label them with some imaging agent, for example, with iron oxide nanoparticles, which we can visualize by imaging method, as is magnetic resonance imaging. Therefore, these engineered EVs can be used for examination of binding of viral mimetics to the receptors by using imaging. And how did we prepare this so-called SARS-CoV-2 mimetics? So we used a plasmid which expressed several proteins on the surface and also inside. We used a system which is based on iron, so it means that the part on the other side of the transmembrane domain stays in the cytoplasm, what consists of green protein together with puromycin resistant. And on this other side of the transmembrane membrane, displayed on the surface, there was a red fluorescent protein and already mentioned RBD. In order to have a versatile system, we also use the alpha tag, which we can use for the testing of this system. So after transfection of the cells with this plasmid, we generated so-called RBD cells. And after isolation of extracellular vesicles from these cells by using differential centrifugation, we isolated so-called SARS-CoV-2 mimetics, or here referred as EV-RBD. As a control construct, we used the plasmid, which expressed the same proteins, but not the RBD. So after transfection of the cells, we created no RBD cells, and the isolated EVs are here referred as EVs no RBD. To confirm the proper expression of these proteins, we also performed the confocal microscopy of these cells. And here you can see an image of a cell with a green protein inside of the cytoplasm. Also, we took a look on the red fluorescent protein, and you can see that it's expressed on the surface of the cells and in the membranes inside of the cell. In order to confirm that this is really an expression on the cell membrane, we label the cells with the label with the dye cell mask, which labels membranes, and you can see the co-registration of this red protein together with the cell mask. The next step was that we tried to characterize the displayed peptide, and for this purpose, we used this already mentioned alpha tag, which was at the end of the RBD peptide, and this allowed us to detect this whole protein RBD fused with alpha tag with a homemade alpha tag nanobody. So we incubated both type of cells, RBD and no-RBD cells with this alpha-tech nanobody. And here you can see the microscopy images. And also you can see that only the cells containing the RBD on the surface, they bounded the alpha-tech nanobody. In the case here, you can also see some fluorescence, the green fluorescence, but this originates from the green protein inside of the cytoplasm. So here you can see also the quantification of this signals. And we also took a look on this system by fluorescent microscopy, by flow cytometry, and at the sickle cell level. And you can see that the difference between the RBD cells and no-RBD cells was very significant. We also took a look on protein expression, and you can see that only the RBD cells contain this alpha-tech. So it means that the RBD is properly displayed on the cells. So by these experiments, we proved that the RBD is properly displayed on the cells. And but this is not enough, because we also want to know whether this protein is functional. So in the next step, we incubated the cells with ACE2 protein, which tell us whether this RBD really binds, whether it has the ability to bind the ACE2 protein. So it was a similar design of the experiment. So we incubated RBD cells and no-RBD cells with recombinant ACE2 protein, which was fluorescently labeled. And here you can see that only the RBD cells bound to this ACE2 protein, in contrast to the no-RBD cells. We also took a look on these cells by flow cytometry on the single cell level. And you can clearly see that only the RBD cells bound to the ACE2 protein. By using this experiment, we can also, by incubating the cells with different concentration of the ACE2 protein, we can also calculate the binding constant. And what was in the case of RBD cells, what was in the case of RBD cells around 6 nanomolar, and the no-RBD cells didn't bind the ACE2 protein. So by these experiments, we proved a strong binding of this engineered RBD to the ACE2 receptors. Upon successful characterization of the cells, we moved to the characterization of engineered EVs. And here you can see the results of the Western blot of the protein expression of protein presence in the EVs. And you can see that they exploit properties of extracellular vesicles. So it means that they contain ALEX and CD81. And only the RBD EVs contain the alpha-tech fused to RBD. We also took a look on the concentration of on the size distribution of the EVs, and it was around 80 to 90 nanometers as it's characterized for EVs. Also here you can see the images from the cryo electron microscopy, and this experiment showed us that in both cases of the EVs without RBD and with RBD, the shape of the EVs was similar, and it was in the range of sizes around 100 nanometer. So by these experiments, we proved the proper size and morphology of the engineered EVs. In the next step, we wanted also to prove that not only the cells bind the ACE2 protein, but also these engineered EVs indeed bind to the ACE2 receptor. For this purpose, we engineered cells to express ACE2 receptors on the surface, and we incubated the EVs fluorescently labeled with these cells. And here you can see that in the case of incubation of the targeted EVs with the ACE2 expressing cells, there was a higher accumulation compared to both, to use of non-targeted EVs or to compare to the hex cells without ACE2 receptors. What proved the binding of the EVs with RBD to the ACE2 receptors. This is also quantified by flow cytometry. And you can clearly see that only in the case of cells expressing ACE2 proteins, they accumulated more of these EVs containing RBD. Upon successful characterization of the cells and the EVs, we moved to the in vivo imaging. We also wanted to show this targeting in vivo. For this purpose, we created an animal model. We injected two types of of cells to induce tumors in mice, in immunodeficient mice. There were HEX cells without just normal HEX cells, and HEX cells overexpressing ACE2 receptors. And after 2-3 weeks, these mice had induced tumors. And an advantage of this model is that you have 2 experimental tumors in one mouse, and you can compare them very, very precisely. So we injected fluorescently labeled EVs into these mice, and we took a look on the accumulation of these EVs in the tumors. And here you can see that only after injection of targeted EVs, there was higher accumulation in the ACE2 expressing tumors compared to the control ones. This was also quantified, and this experiment showed that the accumulation of these targeted EVs is indeed higher in the ACE2 expressing tumors. We also took a look on the biodistribution of these EVs because we wanted to know whether this targeting affects biodistribution, whether it doesn't bind to some other tissues and doesn't have some other effects. And we found out that the biodistribution of both EVs without RBD and EVs with RBD is similar in several organs. So it proved that the targeting does not alter biodistribution of these EVs. Further, we moved to the MRI imaging of these EVs. MRI has some advantages over fluorescence images, what is that we can measure 3-dimensional images. And for this purpose, we labeled EVs with superparamagnetic iron oxide nanoparticles, spions, by incubation of parental cells. And we incubated the parental cells with 4 micrograms of iron per milliliters of medium. And after 24 hours, we isolated EVs from these parental cells. And as you can see here, these EVs were indeed labeled by these iron oxide nanoparticles, as you can see here on the cryo-TEM image. We also characterized these labeled EVs by Western blot for protein expression. And you can see that in both cases of non-labeled and labeled by spions, the EVs have a similar expression of the proteins. Similarly, they also had a similar size distribution and also charge. Then we moved also to the imaging part of the study, that we wanted to see that by labeling of these EVs, we indeed have the contrast on the MRI images. And we performed 2 experiments, one with different concentration of the iron oxides for labeling of the cells, and another experiment with using different concentration of EVs. And you can see here that by both methods, we could clearly detect the labeled EVs in the solution. What proved that these magnetically labeled EVs have a negative T2 contrast on MRI images, and they are trackable by MRI. So after these results, we wanted to also see these EVs by MRI in live mice. And for this purpose, we labeled EVs with the iron oxide nanoparticles, and we injected them in the same animal model containing 2 tumors in the same mouse. And we placed this mouse into an MRI scanner and performed MRI examination. And this is the result before and after injection. Here you can see the tumor tissue, the control one, then ACE2 expressing tissue. And here you cannot really say like what is the difference between these 2 tumors. But when you code this signal into the colors, you can clearly see that in the case of the ACE2 expressing cells, they really accumulated more EVs. This was also quantified by calculation of the contrast to noise ratios. And in order to confirm that indeed this decrease of the signal was caused by iron oxide nanoparticles, we also performed histology examination. And here you can see clearly that the control tumor did not accumulate much iron, in contrast to the ACE2 expressing tissue, where we found several spots of accumulated iron, which is here in blue. So these experiments confirmed the binding of these EVs containing RBD to the ACE2 receptors in vivo. And also because these iron oxides are incorporated inside of these vesicles, it's also proved that we can track the intravesicular cargo delivery by this method. Moreover, I also want to show you the versatility of this method. And because there was during this time, there was pandemic, it was really like increasing, and there was not just like one RBD mutation, but also like the new mutations were coming as Delta and Omicron. And we wanted to know whether we can also assess the binding of these different mutations by our system. So for this purpose, we incubated again the EVs with different type of RBD with the ACE2 expressing cells. And here you can see that by this method, we could estimate the binding efficiency by different mutations. And these results are in line with the already published data, which were published in this paper. And so by this experiment, we proved that this platform can be used also for screening of the new mutations. But here you can see also that the significance is not so high, because Omicron bound the Omicron mutation bound to the ACE2 receptors with a higher affinity, but not significant. But then we also took a look on another mutation which was predicted. It was predicted by our colleague, Jiří Zahradník, and this 62, so-called 62 mutation are supposed to have 1,000 times higher affinity to the ACE2 receptor. So for this purpose, we engineered EVs containing these two mutations, the RBD, the wild-type Wuhan, and the predicted 62 mutation. We engineered EVs to contain these two peptides, and we incubated them again with the hex cells overexpressing ACE2 receptors. And here you can see the results from this experiment, and it's clear that this new mutation, the predicted mutation, accumulated much more EVs compared to the wild-type RBD. So by this experiment, we proved that this platform can be used even for screening of the predicted mutations. So in order to conclude, we developed an MRI-trackable EV-based viral mimetics, which can be used for mapping of a binding of SARS-CoV-2 mimetics to the ACE2 receptors, and it can be applicable also for other mutations or even other viruses by genetic modification. And it can help us to understand the mechanism of viral infections without using infectious material. This system can be also used for delivery of a cargo to the targeting cells, for example, of therapeutics, which were already shown by other group for delivery of siRNA. So I hope I convinced you that by using this system, we can really use EVs as delivery systems, which have very good targeting. Also, they can be visualized and tracked by imaging methods, and they are very versatile because we can, by using the same approach, we can engineer them to express different peptides without big re-engineering of the system. So by this, I would like to thank to all of my colleagues which helped me with this project, also for our collaborators from the Neta Regevsrutsky Lab and Gideon Schreiber. And thank you very much for your attention.
Thank you, Andrea. Beautiful presentation. Please put your questions in the chat if you have any, and I will allow unmuting. If you have any questions, feel free to unmute after I call your name. And I will probably start. What was the size of your iron oxide particles, and how much particles actually you can fit into your mimetic?
So the size of these iron oxide nanoparticles is 35 nanometers, but it's together with the dextran coating. Mm-hmm. And the size of the iron oxide, like inside exactly, it's around 5 nanometers. So when the cells uptake these iron oxide nanoparticles, the sugar, this dextran, helps the uptake into the cell. And then inside of the cells, the dextran is decomposed, and only these small 5 nanometers iron oxides go into the EVs. And regarding the amount, like we didn't calculate exactly like how much iron we have per EV. You could see maybe in this creatine image that there were several crystals inside of one EV.
And was there any like difficulty to kind of pick up a signal, because if you have only a few particles, it's
yeah, exactly. Like it should be mentioned, it should be noted that this method of labeling of the parental cells, it's very low efficiency of the labeling. So not only around like 30% of the EVs are actually labeled. So then also sensitivity of visualization is not that high. So I think this is the main drawback of this study, that we use this labeling. But at the moment, it was also good labeling, because we were sure that these EVs are not contaminated by some excess of the iron oxides, which is usually a case when you label them by, for example, electroporation. But there are already like a new ways how to label EVs, even with iron oxide nanoparticles. For example, they used some tags on the iron oxide nanoparticles, this tag, and then you can wash them the excess of these iron oxide nanoparticles, and the efficiency is much higher, around 90%. Like this was published by another group from Johns Hopkins by Jeff Bolte.
Yes, and we actually have a question from Jeff Bolte.
Sure.
Would you like to unmute yourself?
Sure, I cannot show myself. Hi, Andrea. Hi, Amnon. Good to see you. You remember Jan, Amnon?
Yeah, I want to see you, Jeff.
I cannot on video.
Okay, okay.
We presented this paper at our journal club a while ago. So I have a question for the histology that you showed, Andrea. And you stained for iron using the Prussian blue. In the middle panel at the bottom, I saw all the vesicles kind of in the middle in very large blobs, like that look like fat cells or stroma. And then on the sides were the smaller tumor cells, but they were not there. They seemed to localize in the tumor somewhere, but not in the tumor cells. Or what is that middle band? It was very big and kind of fat. It almost looked like, I don't know, like plant cells or fat cells.
I can go back. Yeah, I agree that it's not localized exactly inside of the cells. If in this image,
if you go back, it's very.
Yeah, I know what you mean. Yeah, here.
Yeah, here.
Actually, I think I'm not a histologist. Maybe it would be good if we would ask someone who is a specialist in this field. But I think that this can be also vessels, like a vessel walls, like endothel, and then the EVs could go into the cells through the endothel. But I'm not really an expert, so I cannot really say. I saw this kind of accumulation in a different part of the tumor. It was not only in this image, and for sure it was more in the case of the ACE2 tumors.
Yeah, it would be cool. We are trying to do now mass spec imaging to see the co-localization of the iron, maybe with the markers of the tumor, but it's very difficult because the amount of EVs that we get in there,
yeah, it may just be that they cannot penetrate really those dense, very dense tumor cells, right? When you see where the size bar is, that they just sort of accumulate in the periphery and near the vascular, but somehow there must be an ACE2 receptor near these endothelial cells then. So, okay, I was just curious about that.
Okay, I have one more question then. Have you tried to do, maybe I missed that in the presentation, but have you tried to do a biodistribution after iron labeling, just to see the tissue distribution of iron, if it's actually binds only to the tumor site?
The distribution of iron? We didn't do this. We did
only something like this. It would be interesting to see if they go someone else as well.
Yeah, for sure. Maybe with this mass spec we could do it. Also with the ICP, but we didn't perform it here. We only took a look on the fluorescence distribution.
Okay.
Yeah, there is a group in Germany that they use isotopes of iron 57, iron oxide nanoparticles with the iron 57, and then you can do mass spec imaging and do biodistribution. But when we injected the nanoparticles themselves, there is no difference between the tumors.
Yeah.
And I just say that we are now using we did one experiment, not in vivo yet, with the method developed by Guanxu Liu here. I think he's here, I saw him before. And hopefully we get better labeling of the EVs, and we'll check it in vivo and we'll go back to you and tell you if it's indeed improve our performances.
Hi, Andrea. Thank you so much for your presentation. I was wondering if you happen to have quantified the efficiency of engineering of your vesicles with the ACE2 binding domains. Do you know the percentage that carry the binding domains, and what's the density of these domains on the surface of the vesicles? And related to this, wouldn't you expect that these vesicles would also relocate to tissues in the mice bodies that are enriched in ACE2 receptor?
Yeah, thank you for the question. Regarding the first question, like we didn't perform this experiment to assess like how much of this we have on the surface. Maybe it's good to know, but we didn't do it or yet. And regarding your second question, you're right that these engineered EVs should bind to the ACE2 receptors, but the mice don't have receptors, ACE2 receptors for this human RBD. So we used, you cannot really like see biodistribution in mice of the ACE2. For this purpose, you can use hamsters, but it's a bit more complicated. We tried it, but we didn't get into the final big group of animals.
Great, thank you.
Andrea, would you like to add something about your study? What will be your future plans then? Like, what the immediate experiments you would like to perform?
Which experiments I would like to perform?
Yeah.
No, I mean, I'm really excited to work more on the imaging part of the EVs, like to increase labeling of the EVs. As Amnon mentioned, we tried already this labeling by the iron oxides, which you can really get rid of the excess of the iron oxides. So to go in this direction, like to really improve the imaging capabilities, maybe to try different contrast agents for labeling of the EVs, different modalities, MR modalities, for example, to track them, to really see them, to really see the effect of them.
I will just add, I just want to add that this system is very modifiable. So you can take the plasmid and just replace the RBD peptide with your peptide of interest. And it's really general approach. Labeling of EVs with the particles, we have to work on it. But in matter of engineering the EVs, I think we optimize it really nicely. And regardless the application, imaging or others, targeting EVs with these methods, I think will be really efficient.
Yeah, we have one more question in the chat box. Gwen, would you like to unmute yourself and ask this question?
Hello. Yes, I was wondering if you managed to calculate the limit of detection for your MRI or SBIO labeled EVs.
So in this case, we didn't calculate the detection limit. I just, what I show in the previous slide, I think we can show you, that I use like a different concentration of the EVs here. And the detection, like this one has 10 power to 11 of EVs per milliliter. So this one has a half of it. So I think this is kind of our detection limit. We really have to have like high concentration of these EVs to have a sufficient signal, around 10 power to 10 or 11, I would say.
And the plasmid, I see something, Jeff, we can send the plasmid, you can try MPI if you want.
Yeah, that would be great because it may be more sensitive and it's quantitative, as you don't have the other dark background as well. But we also need the transduced tumor cells then, right?
We can send you also the cells with the ACE2. I'm sure you have at Hopkins, but yeah.
Yeah.
Or you do it on hamsters, but I don't know. The hamsters are very big. So in our MRI, they are not going in. And they're cute, of course, so you don't want to kill them as you kill mice.
But ideally, we get the vesicles and the tumor cells. Because there may be difference in preparation if we do it here to isolate the EVs.
Yeah, we will meet in 2 weeks. We can discuss that.
Okay.
That's great. We like to establish collaboration during EV clubs. That's the actual purpose of our meetings, right? Okay, great. If we will not have any more questions from participants, I think we can wrap up. Thank you so much for the presentation. It was beautiful. Thank you, and we'll see you next week.
Thank you all.
Bye. Thank you.
Bye-bye.